and I can introduce you to your next speaker, which is Bob from Lavata from the University of Leiden, where he is head of a division for drug discovery and safety in the Center for Drug Research. Okay, sorry, it's a very long title.
It's on the slide here, so
okay,
it's easy.
Too late. Okay, but so Bob is well known for his research over the last almost decades, where he used transcriptomics and different cell models to unravel molecular mechanism of toxicity. And he's also coordinating the EU Tox Risk Consortium, but he's now also coordinating the EU Risk Hunter Consortium. The title of his talk is Breaking Barrier for Toxicogenomics in Risk Assessment, which perfectly fits to my introduction, I guess. So thank you. Thank you for coming.
Thanks for the introduction and also for inviting me here. At first I thought, well, what shall I talk about? More general about the Auspice cluster, the Risk Hunter project, etc. But that's what I do so often, so I thought, well, maybe let's dive a little bit more into the science. Maybe that was the wrong decision, we will find out afterwards. But in all the projects, what we basically use is a lot of the omics approaches to understand mode of action. And if we basically look at the biology and in the context of adversity, you know, the stress response that are taking place early on after exposure to a chemical are really critical. And likely they drive both adaptation as well as adversity. So there's a lot of information in there that may help us to directly address the question, what is the likelihood of a potential adverse outcome of a particular chemical exposure, if you use the right test method, and of course the use omics approach. Now, if you look historically, of course, toxicogenomics is not new. I mean, it started more than 20 years ago. There was a real hype, you know, toxicogenomics will solve all our issues. Particularly in the pharma industry, they invested a lot. Companies were built on it. They failed, got bankrupt. But still, of course, the omics approaches hold promise in understanding what the effect of chemicals is on the biology. And pharma was really asked also by FDA, well, can you put in your dossiers voluntarily omics data, but that really never happened because you can imagine that there's a lot of information that may lead to hypotheses that they have to address. And that's also an issue. How will you deal eventually with all that information that you have? So mostly the omics approaches were used to derive hypotheses on a potential mode of action. Now, in that same period, of course, there was a shift in the technology, moving from spotted arrays now all the way to RNA sequencing. And there's also the question, well, how does the change in technology impact on the conclusion that you can draw? Now, over the years, there has been a consortium, the MAQC consortium, that has tackled most of these technical issues. Now, is the technology fit for purpose? Is it robust? Can you get the same answer independent of the technology? And that has all been published over the years, in the past 20 years basically, by this consortium. At the same time, if you are an individual researcher, then the question is, well, how do I report my information? Because there may be variability in how you report it. So in particular, there has been proposals, guidelines that has been suggested, particular on reporting the design of the toxicogenomics experiment, as well as the bioinformatics approaches, but not in the end on the interpretation of the data. And that's of course key in a risk assessment. You want to understand, you know, what does the data really mean? How can we interpret the data, and can we make it quantitative to make an ultimate decision in the context of a risk assessment? So there's a current gap that we have in a framework to reported information. Now, one of the hurdles in this interpretation, both from a qualitative as well as quantitative aspect, is that, you know, a lot of information on gene, the role of individual genes is from cancer biology. Those have been the major drivers in understanding mechanisms of action of individual genes. So that's the biology we're referring to. So that's one point. Then the commercial as well as public domain platforms that integrate omics data, they make use of that information. So what you get back is basically input. So there's a lot of redundant information on pathways that mostly relate to sort of cancer biology programs. Not only, but for a large extent. So that limits us in the understanding that real biology that's ongoing. Secondly, all that information is based on all the knowledge that we have in the public domain, but not on a particular test system you have at hand. So you may give a wrong interpretation of your results, basically. And thirdly, the statistical method and the variability in your assay may determine what type of genes you identify that may be different by different researchers. So all those problems need actually a solution, and I will explain the approaches that we actually take in the different projects that we're involved from that perspective of the interpretation. At the same time, to derive a point of departure, you need concentration responses, and in the past, and still RNA-seq, individual full genome RNA-seq sequencing is still expensive. Which limits, of course, the amount of samples you can take to get a dose response. And therefore, we now have a solution that is taken up both in the US as well as in Europe, using high throughput transcriptomics approaches, using targeted RNA sequencing that reduce the cost and allows basically to get really dose response information. Now and that relates to the first solution, that interpretation, we take the approach of looking at the biology where transcription factors act on genes because they want to respond to the particular perturbation. And these transcription factors act on gene sets, and depending on the perturbation, they need to do something as a network that is regulated by transcription factors. So what we try to do now is build actually these co-regulated gene networks in a particular test systems to really understand what is the underlying biology in a test system. And a test system can be a full organ, a liver or a kidney. It can be an in vitro test system, primary hepatocytes, or other test systems as well, and I come back to that later. So what you try to do is actually link over a large data set, preferably a few thousand individual experiments, you try to see which genes are now always co-expressed independent of the experiment. So that's preferably, see if I can have the laser here, that's particular over here. Here you see a lot of experiments where you see co-regulation of the genes. So you seek genes that always are co-regulated, that will become a network. So if you have sufficient data, and there's a lot of data in the TG Gates data set, where you can do such experiments and really use this data now to see which genes always are co-acting, and are they now in a network? And can we understand the biology in a network, but also relate those networks to a particular type of biology? So that is what we have been doing, using the TG Gates data set, and actually build now these co-regulated gene networks and build maps of those. So what you see actually in the images over here are individual maps for example, for primary hepatocytes, or for the liver, or for the kidney. So this is, of course, in vivo. This is in vitro. And each individual dot, what you see here, is a gene network that integrates genes. And I'll come back to that later. Now, visually, it's very easy. You know, you see a map, you see where there's activity. And if you understand your map, it's just if you see a map of Europe, and you need to go to Leiden, you know where it is, it's in the Netherlands. And the same here, if you see a particular activity in the map, you know if you're knowledgeable, this activity represents something else. But you also can compare compounds, of course. I'll come back to that later as well. Now, we built that map in the context of a platform where you can really, as a biologist, and I'm more a cell biology and toxicologist, I'm not a bioinformatician, so I cannot use all the data, but I can play with this tool and really start integrating the information that I have from the toxicogenomics data set. Importantly, if there's also information on pathology, so there's also pathology associations from in vivo data, you can also start linking gene networks and their relationship to a pathology. and therefore get actually biomarkers of an adverse event. And that's what we have been doing as well. Now, how does it now look like? So this is a particular exposure situation from in vivo, 15 days, cisplatin treatment, and then you get the transcriptomics data, and this is the map you have. So you see activities, and you see a strong activity over here, and you want to explore what it is. So what you do the next is you look into detail in such a gene network and start asking the question, well, what is that network doing? Are the genes in there? What are they? Are they active? Yes or no? And is the activity of individual genes, is that also represented by the activity of the gene network? So you get a quantitative score of gene network activity. But in each gene network, you can also relate, well, what does it represent? Are there transcription factors that regulate the network? Are there gene ontology terms that say something about the biology of that network? So it's very intuitive to work with a tool and explore basically the biology. What you can also do is start comparing your particular compound with the data that's available and see which particular compound is looking like cisplatin. Now in this case, we have puromycin. You see a strong correlation. So these are scores for gene network activities. So here you see all the gene network and their activities. There's a strong correlation. There's something over here, and that is representing basically p53 cycling. So the DNA damage you see in cisplatin, but you don't see it in puromycin, but other programs are similarly active. So you can really explore now the different biologies from two different compounds and get understanding if they're really similar, yes or no, and to what level. Now, you can say, well, that's all nice TGH data, but if you now take a new data set with the same compound, would you get something similar? How robust is the system? And do you see particular mode of actions? Do you see that back not only in one target organ, but you see the same mode of action of another target organ? So in this case, we looked at cyclosporin A in another project, an IMI project with pharma industry. We had an individual data set, almost similar dose, similar treatment, but also now using TempoSeq, and here it was Affymetric gene array. So first of all, would you get the same activities with the same compound, but also do the same different platforms give you the same result? And that's what you actually see here, that if you compare now the liver map for these treatments from this experiment and this experiment, you see that there's a high activity at similar nodes. So that really helps you to understand now, is the system robust, yes or no? But you also see now that if you look at the kidney, the activity in kidney is here not so strong as over here for the same dose, but the biology is similar. So this biology is in both cases ER stress signaling. Now, can we apply that now? So we also have this primary hepatocyte TGIC mapper tool, and what we actually did, we applied it in the context of a case study in EU ToxRisk project, where the question was really, can we use read-across approaches supported with biological information to get a read-across for VP analog? This case study was really driven by Sylvia Escher. We reported a lot of the data from different NAMs in a report which was evaluated by the OECD as a case study report. But what he didn't include at that time was the transcriptomic information. So can transcriptomics analysis, can that contribute now to the evaluation of a similar mode of action at the biological level? So that was the question that we actually addressed. Now we have this PHH mapper tool, and what you actually see now, if you treat these primary hepatocytes with VPA in vitro, then you get an activity. Now if you remember the other map, that looks different, because then there was an activity over here with cyclosporin A. Now you have different modules being active. The question is then, okay, What are those modules? Do they make sense in the context of a steatosis onset? And if you look now at this module that is strongest active, that is module 31, each module has a number, and then each module has a link to the biology. So if you look at the strongest module, that is actually related to fatty acid oxidation, which is related to steatosis. So that module links basically to what VPA is doing in the context of onset of steatosis. Now, we had multiple compounds in that case study. We screened a total of 18 different molecules in the dose response. And then we asked the question, well, can we use now this module activity to see whether these compounds have a similar mode of action? And that's what you basically see in this slide. So here we have valproic acid at 60 millimolar, so quite high. We did the full dose response. I'll come back to that later. And then there's another compound that belongs to the same class, similar dose, and you see that there's a very strong correlation on the activity of the modules. At the same time, you have compounds where there's no correlation at all. So you can now basically make a matrix of all the compounds you have and look for the correlation efficient in the activity of all the modules. So it's very simple. You don't look at all individual genes now. Now you look at the overall activity of the biology between two different compounds. And what you now see is that there's a cluster of compounds that has a similar activity. They're really correlating in their mode of action at this dose of 16 millimolar. But you can expand it and basically take all the concentration you had in the experiment and get a big matrix, but that's very diffuse on the screen, get a big matrix on what concentrations are now really relating together in this mode of action. Now, I mentioned this module 31 that is related to this fatty acid oxidation, and I don't have it here, but this module was the one that was really active at already very low concentrations. So in the biology, a module that was activated very potently. So we thought, well, given that this module is actually related to the adverse outcome, maybe we can design actually also approaches to start defining those responses on the activity of this particular module and use that actually as a way to derive benchmark concentrations for the potency of the different compounds. So that's basically what you see here for all the compounds. You see over here valproic acid. It's already active, starting to be active at low concentrations. And in the dose response, it then activates this module 31. But other compounds are completely negative. So this allows us now to actually use such an approach in the read across, not only to look at comparability in the activity, so that's basically what we did here, but also in their potency. Yeah? So we can derive potency insights on the mode of action of these molecules. Now, you can then say, okay, that's all nice. You use primary hepatocytes. They're costly. Everybody wants to use HepRG because they're more easy to use, or they want to use something else. The question is then, of course, would you see the same in HepRGs? Or are different test systems, will they give you a different response? So we did a large study in a Cosmetics Europe project where we now had the ability to really look at time course as well as dose response. So we can now derive not only information between two test systems, so HepRG compared to primary hepatocytes, but also whether the biology is actually similar on different time points. So how robust is actually your testing strategy? Now, if you look at this graph, so these are just the differential expressed gene numbers. You said, well, it's sort of comparable for valproic acid. So we could use VPA, or we could use HepRG cells maybe for any test system. Well, maybe for VPA. We also tested APAP and cyclosporin A, and here you see clearly a difference. So now in the potency for inducing gene set changes, you see that HEPR-G is not so sensitive for APAP, despite the fact that there's metabolism there. Yeah. For cyclosporin A, it's even worse. So the dose response, we only see dramatic changes at high concentration, but we do not really get a concentration response. Now, if you would be a toxicologist, you would have spent a lot of money on nothing, because if you paid for all the samples, there's no effect. So these experiments are very costly in a way, without getting maybe a lot of data. And that's part of the strategy that you need to accept, of course. But you see here now that depending on your test system, you may get different answers. So we need better evaluation of test systems and what is fit for purpose and what's not fit for purpose. But of course, we applied these data sets now to this TEG mapper, and then derived for the various modules that were active, derived these benchmark concentrations, and then compared basically the test systems. Now, this is what you see in this graph over here. It's very detailed. In essence, what we have is on the top modules that are very active overall, and the bottom less active modules at different time points. So, for example, you would see for cyclosporin A that the top module is active already at early concentrations, whereas other biologies are only getting active at later time points. Now, what we actually can see is that the BMC value you derive is dependent on the pathway you have. So depending on the biological change, you may have a different BMC value. That in HEPR-G, we see similar activities for VPA. So the biology here is quite similar between the two test systems. So apparently, the impact of VPA on HEPR-G and primary hepatocytes is similar in their biology, which is of course relevant because it demonstrates that HEPR-G represents biology that you also see in primary hepatocytes. But then what we also see, and I mentioned that before, we tested cyclosporin A in vivo, we see our ER stress. In vitro, we see basically the same biology being active. So that's comfortable in relating in vitro biology with in vivo biology. So what is maybe a larger problem is that primary hepatocytes are more sensitive for HEPR-G. Now you can have the discussion, yeah, that's because you have isolated them, they are more sensitive, that's what you expect, or is HEPR-G actually a poorer model than primary hepatocytes? And that's of course a question that we cannot answer. We can provide hypotheses, but that's what we have to deal with. So I demonstrated now, a tool, an approach to easily interpret and quantify toxicogenomics data. We have applied it in different case studies, but we are aiming now for in different projects with an ultimate vision that we would have actually such a tool for different test systems. So we have red liver, I demonstrated that. We have primary hepatocytes now. In the Risk Hunter project, we have it for HepG2 cells. But we would like to see now that such a pattern also in different liver test systems, and then understand, is the biology now between these test systems similar? And do that also for kidney, for lung, for neuronal systems. Also being able to see is what type of biology is now more similar between liver and kidney and the other target organs. And that helps us actually to better define how we can use toxicogenomics data, interpret the data in a quantitative way, but importantly also translate it to a human setting. So derive also human samples, rather from pathology, also derive these models and see now what type of biology in the test systems do we see back in a human liver in vivo. And that will provide ultimately confidence in how we can use these approaches in a risk assessment setting without using animals, despite the fact that we can also relate it to the red liver, but we want to, of course, relate it to the human liver. Now, George already mentioned this project that we will start, the TD-Track project, because here we will now also look at the inter-individual variability, but actually derive now also TG Mapper of PBMCs that are derived directly from the patients being exposed, and that might allow also applications for biomonitoring in the future. Now, to extend this, we built a map. What's important, we want to connect it to pathology and cell diversity information, anchor these model activities to AOPs. So can we use it to quantitate AOPs as well? Also related to, of course, defining point of departures, really for risk assessment. And the application I already demonstrated for read across. And we have to see how we can apply it also for ab initio approaches. And that's where I want to stop, thanking, of course, our funding agencies and the people that did the work in this particular presentation.